Today we start chapter 3, which is about enzymes, and this is the biology syllabus 9700 for A levels biology. This chapter is the third chapter in the AS level syllabus or for the A1 syllabus. Now basically we're going to study enzymes and how they function. The first and the foremost thing which I like to discuss with you all is define active site. Now most of you are not unable to define it, and you give me all sorts of descriptions about it, and now I did not draw an enzyme like this, and I did not say that this was the part where the active site. So you can see this enzyme X is very different from enzyme A, B and C. The reason is that if we say we had to define active site, so active site is this part, and the active site is actually a depression. It's a depression or a cleft. And I usually say the active site is like a khadda in which the substrate fits. So it is not just, you can't say an area where the substrate fits. So it is a depression, it's a sort of a cleft. The word depression or cleft has to be used. Now you can see how enzyme A, B and C are totally different. The shape of the active site is different. So important that we use the correct biological English, the shape of the active site. So the shape of the active site is different in these three enzymes. So different substrate would fit, and we say the substrate, the shape of the substrate is complementary to the active site, or the active site, the shape of the active site is complementary to the substrate. So we have to be very clear about what we mean by when we say active site. So active site is different, so that is why A, B and C are three different enzymes. Now these three enzymes would be coded by three different genes. And so gene A would be for the enzyme A, and gene B for the enzyme B, and gene C for the enzyme C. Now how are they different is because they would have a different base sequence in the DNA. Now these base sequences will code for the sequence of amino acids. And that means the primary structure of these three enzymes would be different. So we know enzymes are proteins. So proteins are made up of amino acids. Now how is it possible that three different enzymes have three different active sites? The reason is in the DNA there would be different base sequences which code for these enzymes, enzyme A, enzyme B and enzyme C. So the enzyme A would have a different primary structure, and that would result in that enzyme having a specific shape of the active site. So of course this is to do with the DNA chapter, but I want to be able to tell you this now so that you can understand it, and then we can apply this knowledge later on in chapter 6. Now we say enzymes are globular proteins. So number one, globular protein means they are round, or you could use the word spherical. And the main thing which we have to understand is that the amino acids have the hydrophilic R groups on the outside. So the ones on the outside are the amino acids with the hydrophilic R groups. And the one on the inside are the hydrophobic R groups. Now that's a very broad definition of globular proteins. Now this is very important because it has to be water soluble. Enzymes are all working in a very watery medium. So globular proteins, number one, round. Number two, you can use the word spherical, or you can say ball shaped. And they have to be, the important thing is the hydrophilic R groups are on the outside and the hydrophobic R groups are on the inside. Enzymes are of two types, intracellular and extracellular. Basically, intracellular enzymes will be made on the free ribosomes inside the cell. So the free ribosomes are present in the cytoplasm are the ones which are going to make the intracellular enzymes. Now the intracellular enzymes are those enzymes which work inside the cell. They work inside the cell. Now the extracellular enzymes are the one which are made on the fixed ribosomes. Now the fixed ribosomes are the one which are present on the RER. Now what we have to understand is the fixed ribosomes are also ATS. Now we are talking of eukaryotes. So we are talking of eukaryotes, and the ATS ribosome present, free and fixed, both are ATS. The fixed ribosomes are going to make extracellular enzymes. Now if you remember in your O levels, we talked of the stomach, and in the stomach, the cells of the stomach wall make an enzyme, and that enzyme is called pepsin. Now this pepsin works in the stomach cavity and it digests the food that you have eaten. So whatever food you have eaten, if you've had some chicken or if you've had some meat or if you've had some fish, well, that protein in your diet is going to be digested. So the enzyme pepsin, which was made inside the cell, is going to be thrown into the cavity of the stomach. So the enzyme pepsin, this enzyme pepsin is an extracellular enzyme. Though it is made inside the cells of the stomach wall, but it works outside the cell. It works in the lumen or the cavity of the stomach. This is the stomach cavity where the food goes. So this is called an extracellular enzyme, and the extracellular enzymes are made on the fixed ribosomes. So intracellular and extracellular enzymes and fixed and free ribosomes. The enzyme and the substrate combine to form an enzyme substrate complex. That's also called in short ESC. So enzyme substrate complex, and then the enzyme is again reused, and what is the final? It could be one product or it could be products, because one molecule, like we have starch converted by amylase into maltose. So what you need to understand is that the enzyme combines with a substrate and forms an enzyme substrate complex, and then the enzyme is free to work again, so it's sort of reused or recycled. And then what is formed out of the substrate, either the substrate was a large molecule and then it broke up into two, so it would be two products, or it could be the other way around. This could be the substrate and it could be combining it up and making it into one product. So enzyme substrate complex and then enzyme product or products. Then when you look at the basic understanding of enzymes, we say enzymes lower the activation energy. Now what is activation energy? Activation energy is the energy which is required for the substrate molecule. So this is the energy level of the substrate molecules. Now they need all this energy. So we say, if I had to draw a graph, and this was the progress of the reaction, and we had energy on the y-axis. Now this is the energy of the substrate, say it's 50. Now we need to increase, this is called the activation energy, so we need to give it up to 100, and then the reaction proceeds and the products are formed. So this would be the substrate at this level. Now we say this is the activation energy. So 100 minus 50 is the activation energy. Now this is without an enzyme, but when we say an enzyme is used, then we say the enzymes lower the activation energy. Now basically the energy level of the product and the substrate must remain the same. So what has happened is that the energy is now reduced. So the fact that we say the enzymes lower the activation energy, now this has to be very clear to everybody that this is now the energy level which was needed. Previously, this was the energy level which was needed, so we needed this much energy. So we say enzymes lower the activation energy. So enzymes will result in the reaction proceeding very fast. So the reaction proceeds fast and we get the products. Now all enzyme reactions are actually very, very important in biology because animal and plant cells all depend on enzymes. So we say enzymes lower the activation energy. Then we have two hypotheses. One is called the lock and key hypothesis and the other is called the induced fit hypothesis. Now in the lock and key, it's very simple. We just say, the enzyme, the active site of the enzyme acts as the lock. So the enzyme is the lock and substrate is the key. So the substrate acts as the key and they just fit in it, and we say the shape of the active site is complementary to the substrate, or the shape of the substrate is complementary to the active site, and it fits in and it catalyzes the reactions. Temporary hydrogen bond forms between the R groups of the active site and the substrate, and the reaction is catalyzed and it breaks up into two, and that would be the products. But in the induced fit model, we have a slightly different view. We think that the enzyme is there, right? It's got an active site, but the substrate sort of fits into it, not very accurately. And then we say the enzyme sort of molds around the substrate. So the shape of the enzyme changes. The shape of the enzyme changes and we say the enzyme molds around the substrate. Molds around the substrate. In the lock and key, we did not say this. In the lock and key, we did not say this. We just said that the shape of the substrate is complementary, so it fits into the active site. But in the induced fit model, we say as it says here, in the induced fit model, the enzyme undergoes a conformational change upon binding to the substrate. The shape of the active site becomes complementary to the shape of the substrate only after the substrate binds to the enzyme. While in the lock and key model, the shape of the substrate and the conformation of the active site are complementary to one another. So these are the two hypotheses which we still keep on dwelling about, and some biologists prefer one and some biologists prefer the other. So we've got to understand is this is an hypothesis. It's not been as yet proved or disproved, any of it. So the lock and key model and the induced fit model, these are the two that we need to discuss for this chapter on enzymes. Please pause the video and make your own notes and then go through the video 1 or 2 times, and then you can start doing the questions on it. And now as we go along the syllabus, we come to 3.13. Now it says investigate the progress of enzyme controlled reactions. So how do we investigate the progress of enzyme controlled reaction? Now how can I judge your progress in your A-level biology? Well, I can take a test, and maybe 2 weeks after we started the course we can take a test, or we can do a test after every chapter, and I can study your progress. I mean, if you're getting 75% marks in every test, well, you're doing very well. But if you're getting 50% and then sometimes you're getting 30%, well, you're not doing very well because some chapters are not very clear to you. So how do we study the progress of enzyme controlled reactions? Well, we have two methods. Now, first of all, let's discuss a very important enzyme reaction, which is starch, and we're studying things at O levels. Amylase converts it into maltose. Now, maltose is the product and amylase is the enzyme and starch is the substrate. Now either we can study this is if we can, now the first method is the rate of formation of product. So if we can study, okay, the enzyme maltase was 0 and then in 5 minutes it is 100, then in another 5 minutes it is 1000 and it stays 1000, which means that that is all what the starch was has been converted to maltose. So one is the rate of formation of product. Sometimes we can do that, sometimes we cannot do that. The product, if it's oxygen, like in the enzyme catalase, that of course can be measured because we can measure the volume of oxygen produced per unit time. Or we can measure the volume of oxygen after say 5 minutes or 10 minutes, and when it doesn't increase anymore, that means all the starch has been used up. The second is if we can study the rate in which it takes, how much time. Please remember, rates is always time. Always rates, it's time. Whenever we have to study rate, we're talking about time. So if we can study, okay, the starch was 1000. How much time does it take to become 0? So it takes 5 minutes. So that means the rate of disappearance of the substrate. How much time did it take for all the substrate to be converted to 0? Like for instance, as I just said, it was 1000. We knew the starch was 1000. And then it took say 3 minutes and then it was 0. Now we can use the iodine test to do that, to find out if the starch is present. And we can, after every 30 seconds, maybe take a sample, which I'm just going to discuss with you in the practicals. So this is 3.13. Investigate the progress of enzyme catalyzed reactions by measuring rates of formation of product, or the second method is rates of disappearance of substrate. And of course we're going to be discussing, I've talked about amylase and we've talked about starch being converted to maltose, or the enzyme maltase, converting the maltose to glucose. So if we can measure the glucose, then that would be the rate of formation of product. If we are going to take, okay, how much time does it take for the maltose to disappear? Well, that will be rate of disappearance of the substrate in the enzyme controlled reaction in which we are talking of maltase. Topic is investigate and explain the effect of temperature on the rate of enzyme reaction. Now I've drawn a graph, and on the x-axis we have temperature in degrees Celsius, and on the y-axis we have rate of enzyme action, and that's in arbitrary units. Arbitrary units means that it's just some figures that I have given, and I've given arbitrary units that maybe 100 product per unit time is 10 or 15 or 5 or 10, 20. So arbitrary units means some figures which is there's no proper unit to it, but just something which you have assigned a figure to. So effect of temperature on the rate of enzyme reactions, you see every enzyme has an optimum. Optimum means the temperature at which it works best. Now every enzyme, human enzymes have an optimum of 37. But there are bacteria which survive in volcanoes, and those could have a very higher optimum temperature than their plants which survive on glaciers. They could have a very low optimum temperature. So optimum temperature is very variable. And every enzyme will have its own optimum. So you can't say all enzymes have an optimum temperature of 37, because that's not correct. So all enzymes will have their own optimum temperature at which they work best. Now this graph that I've given you, between A and B the rate is increasing. So between A and B you can see the rate is increasing, and then as the temperature after B increases, the rate starts to drop, but it doesn't drop to 0 immediately. It still takes some time before it is totally denatured. So if we read off the B at the point B, the temperature is probably 18 degrees Celsius, and that would be the optimum. So before the optimum, for every 10 degree rise, and that is called Q10, is equal to 2. Now you can see at 5 the rate was 10, at 15 the rate is 20. So anything 20 over 10 will be 2. So if for every 10 degree rise in temperature before the optimum, the enzyme coefficient Q10 is equal to 2. But after the optimum, you can see what is happening is that the rate is decreasing, and the rate is 0 at C, in which the enzyme now is totally denatured. Now between B and C we say partial denaturation, because now the product is being formed but the rate is decreasing. So this is called partial denaturation. So A and B, the rate is increasing, B and C, the rate is decreasing. But then of course there's an MCQ of yours that where is the kinetic energy the highest? Is it A, is it B, is it C? Where is the kinetic energy the highest? Is it A, is it B, is it C? And mostly you say B, but then you're not reading the question. The C is the highest temperature. B is 18, C is 30. So C is where the kinetic energy is the highest, and there's an MCQ on that. Now as you look at this graph, there's a very typical question which comes is describe A to B. Describe A to B, and then the other question is explain A to B. Now they're usually two very different questions and you need to really understand. Now the first and the most important thing is that describe is no biology, and you just have to describe the graph. So in describe you usually just give figures, so we'll say at 5 degrees Celsius, and you must give the units. The rate was 5 arbitrary units. Then at 15 degrees Celsius the rate was 20 arbitrary units. So it's just figures, and you just have to say increasing temperature increases rate. So as we increase the temperature, the rate also increases. Rate also increases. So this is what we have to understand, is that when we are writing a describe, we are not going to give any explanations. An explanation is a totally different thing. So describe A to B would just be figures which you give. You must give the units, and you just give me the rate. So you just read off the x-axis and you read off the y-axis, and you give me the figures for that. And that usually is, everybody should be getting those marks. So for instance, the describe is for 2 marks. So there should be nobody who misses out these 2 marks. Now the explain is where you give me biology, is that you know increasing the temperature. As we increase the temperature, this increases the kinetic energy, and as it increases the kinetic energy, more collisions, more enzyme substrate complex is formed, more ESC, enzyme substrate complex formed, and more product per unit time. So more, now this is biology. So if I say explain A to B, you've got to give me something which is, so in the same question I've sort of given you the describe and the explain. So describe is just giving me things of the graph which everybody can do, and you, even if you're not a biology student, you can do it. But when I say explain, then that's only a biology student can do that, because he or she only knows what the explanation is. The explanation is that increasing temperature, kinetic energy increases, more collisions, more enzyme, more product per unit time. Now this of course is going to be the explanation, and that is maybe this is for 2 marks and this is for another 2 marks. But if you have written this here and you've written this here, well, you get a 0, because you don't know what a describe and an explain is. So be very careful when you are answering these questions in the exams.